Hi, Mansi.
Yeah, hi. So sorry, you must have panicked that I was late. I was just running
It's okay.
another
Yeah.
thing.
So we'll start in a minute or two so that
Yeah, yeah,
people
that
are waiting. Yeah, sure.
okay. Yeah, thanks. Amrita, I'll just share my screen once.
Yeah,
Yeah,
sure. Yes.
I think you have disabled share for
Uh,
me.
Wait. Share screen. Yeah. Can you please check now?
Yeah.
Yeah. No.
Uh,
Yes. Yeah. Yeah. I can see the. I
it's
can.
not full screen yet, right?
No. It's not on slideshow. Yeah.
If I d-
Yes. Can you please move the slides so that? Yes. Great.
yeah.
The size. But the size is
The size
getting
is an
small.
issue. Let me just
I think that is because it is PDF.
let me no, this will not work. Let me share the entire screen.
Yeah, works.
Let me just use another 1 second.
Sure, no problem. Meanwhile, I can start with the introduction. So, welcome everyone. So today's week 10 live session on macroecology and case study. So here we have Mansi Mungi for the week 10 guest live lecture. She is working as assistant professor from in Azim Premji University, and she did postdoc in the University of Leeds, UK. Her expertise are community ecology, macroecology, policy making, biodiversity, and conservation. She did her PhD from IISER Pune on patterns in diversity and community assemblage of hawk moths and birds. Uh, yeah, so let's hear about macroecology case study, which she's going to take live lecture today. So you can ask questions in the live chat. So she'll be answering those questions after the session. I can see some of them have already asked questions, but all those questions will be answered Mansi after the session. Yeah. So thank you everyone for joining. And yeah, now I can see the full screen. Yeah.
Okay,
Over
excellent.
to
Yeah.
you, Mansi. Yeah.
Thanks, Amrita. Will I be able to see the questions? No, I don't think I can see them.
I can copy paste it to the live chat here.
Okay, fine. Great. Huh.
Yes, after the
Thanks.
session. Yeah.
Okay, so I can start now.
Sure,
Right.
sure.
Yeah.
Yeah.
Okay, great. Good evening, everyone. Hi, my name is Mansi, and I am talking today about community assembly of East Himalayan birds, which basically means how are communities of birds structured across the Eastern Himalayas. Now it's a little more nuanced than just structured. Uh, I want to just tease apart the word community assembly first a little bit because it it actually outlines everything else that I will be talking about in this lecture. So in ecology, communities are essentially a group of organisms that can be at any given taxonomic level, which means it can be a community of birds, it can be a community of just passerines, it can be a community of just parrots, right? So you can have a community which is a group of species living together in a defined area and interacting with each other, but you can define that group at any taxonomic level. It can be order or family or a genus, right? So here I am specifically talking about community of birds, which is all birds, right? So my community is defined at the level of a class, the class AVs. Assembly, right? So assembly is simply the process of putting together things with some common predefined purpose, right? So here we are talking about the processes which have put these birds together in the Eastern Himalaya, right? And this field in ecology talks about what processes determine the composition of communities. Why are these birds found in Delhi and not in Kerala, for instance? What determines the difference in the species composition of Delhi versus Kerala in terms of birds? So those are the questions that I'm asking for the Eastern Himalayas. Why are these birds found in Eastern Himalayas and not in the Western Himalaya, for instance? Without any background or a technical understanding of community assembly processes, I think we can all intuitively guess what these processes should be like, right? So to answer a question such as why are these birds found here, it's very intuitive for me to guess why, right? It could be the kind of resources that are found there that determine the kind of birds that will be found there. It can be the kind of environment, temperature, precipitation, the kind of forest, right? So environment is of course the first logical thing that one can guess that determines what kind of species will live somewhere. And the other is it could be the the other kind of species that are found there, right? Because a species can't live in isolation, it will interact with other species. It has relationships such as competition or mutualism, right? It depends on other species for its own survival. So, broadly, these two different categories of processes are called abiotic and biotic processes, right? That together jointly determine what kind of species will be found at a particular location. And yeah, in community assembly, we classify these two into different sort of smaller categories. One is called environmental filtering, and the other is called interspecific competition. So these are a little similar to the biotic and abiotic that I talked about. And I will just spend some time explaining these to you because I want to talk about some of these in more details eventually. So here I have shown a schematic of a single isolated mountain system, and I am trying to understand. So across this mountain system, across a broad elevational gradient, at different elevations, you will have different communities of birds, right? The communities found at the lowest elevations will not be the same as those which are alpine birds, for instance, right? So the environment definitely determines what kind of species are going to be found at which elevation. And if I take together all the species across the entire elevational gradient, what I get is something that we call a regional species pool that is shown in the outermost margin here in this schematic, right? So you have a regional species pool, which are all the species that are found along a mountain, which then get filtered down to a small local community. Okay. This local community is a subset of the regional
Excuse
species.
me, I think the slides are not moving. We are left with the first slide till now.
Ah, I see. Okay.
Oh, now it is come.
Oh, now it is coming. Oh, the slideshow was not moving. Let me. Is this view okay though? Like, can you see the
Yeah,
full
view
slide?
is fine.
Okay,
view
then I'll
slide.
probably just scroll if that's simpler. So
Okay,
you
all
probably
right.
missed this slide in between. This is where I
This is the
talked
one,
about.
all the time we had this,
Oh, you had this.
only this.
Yeah.
I see. Okay, so this is the other one.
Yeah, okay. Thank you.
Okay, thanks. Yeah. So this is the schematic that I was talking about. So to study how a community is assembled, I first take all the birds that are available to form that community, which is the regional species pool. So if I take the example of a mountain system and I am interested in studying what are the processes that determine the community of birds found at middle elevations, right? So I first consider all the species that are found across all elevations, which is my regional species pool. And now there will be the set of processes that will filter the regional species pool and eventually lead to a local community composition. And these processes, these filters, if you may, uh, can be broadly categorized into these two categories: the environment and the interspecies competition. And it is convenient to think of these processes as sequential. And I'll tell you what I mean by that. So imagine here I have shown this all these different species that are found across a mountain system. Now the environment first selects a subset of these species. And these are the species that have the traits that are most suited for the resources available at middle elevations, right? So first the environment will automatically filter out the species that cannot live there. So these are going to be species which are adapted to the highest of elevations or warmer conditions at the lower elevations, right? So these are species that are environmentally suited to live there. But usually, all species which are environmentally suited to live at middle elevations will not be found there. Some of them will be filtered out by competition between those species. Because all the species that can live in a given habitat, it means they all have very similar requirements, right? They are all adapted to that place. So they are all going to compete fiercely for the resources found there. So this second filter again subsets those species. And ultimately, the species that you see at a given location are a trade off between these two processes. And these are the most, there are, of course, other processes. I'm simplifying things a little bit here. But these are the two processes that I had studied extensively during my PhD. And they are also sort of the most two broadest categories that you can have. You can have more processes within them, but these are the two broad categories that you can study. Right. Now, assuming that your local communities are a result of two sets of processes in nature, how do you actually test for the presence of these processes, right? If I go and I look at a community of birds in nature, in the forest, what can I say about, which is more important? Is environment more important for these communities, or has competition played a major role? I know that they are both going to play some role or the other, but I am interested in finding out which is more important, and are they too important at all? How do I study some of these things actually empirically in nature? So for that, I'm going to take a side step for a while, and I'm going to talk about how do we actually quantify communities. Communities are not just a checklist of species, right? When I say a community of birds, it is not just the species of birds found there and the abundances of these species. There are multiple ways in which communities can be actually quantified, measured, or described. And some of these ways are actually very useful to study the community assembly processes like environmental filtering and competition that I talked about. So, three of these metrics that can be used to describe a community or quantify a community are species diversity, which is, of course, all the different species that are found. But the other two are phylogenetic diversity and functional diversity. I'm not sure if these topics have already been covered, but don't worry, I'll just quickly explain some of these because these are very important concepts that help you understand the nuances of how to describe a community. So I'll talk about functional diversity first. Right? So here I have shown two different communities. One is a community. Can you see my cursor? I'm not sure. I'll take my chances. Okay. So this is a community of parrots, and this is a community of birds from multiple different orders, right? Both of these have 20 species, right? So if I have to compare these two communities, I can compare them using species diversity or species richness to simplify things. And I will say that both communities are equally rich in terms of the number of species because they both have 20 species. But now, if I go and measure something else about this community, not the type of species, but let's say a particular trait, right? So I say that, okay, instead of the type of species, let me measure the beak sizes, right? So I can measure the beak sizes of 20 parrots here, and I can measure the beak sizes of all these birds here. And I can look at the distribution of beak sizes. And I will see if you compare these two graphs. So here is the distribution of beak sizes of parrots. So on the x-axis, you see the beak size. Okay. And all these values, this is of course a hypothetical example. So you can see that it's a very narrow distribution. That's because all parrots have very similar beak sizes, the beak volumes, right? They're closely related to each other. They have similar eating habits, right? So they are all functionally very related to each other. Whereas if you look at the other community, you have a wider range of beak sizes here because we have beak of an ibis, which is slender and long, versus the beak of a parrot, which is, curved. So when instead of quantifying the number of species, I quantify a functional attribute, for example, the beak here, I automatically get a very different picture. I see that this parrot community is actually functionally very poor, whereas this is functionally very rich. There's a wide range of beak sizes available there. And why this is important is because now the beak sizes actually tell me how, in what ways is this particular community interacting with other animals, other resources, plants, seeds, fruits, insects? What kind of items is it eating? What kind of resources is it exploiting? So it tells me about the functionality of that community with respect to its ecosystem or surrounding, right? So I can quantify a community based on just the number of species or in terms of its functions. A community which performs more functions can be considered more important from a conservation point of view. And that is the rationale why sometimes communities are not just measured in terms of whether they are species rich or no, but also in terms of whether they are functionally rich or no. I've taken the example of beak here, but you can do this with body size or wing length or the length of the tarsus, any different number of traits, right? The other thing is phylogenetic diversity that I talked about, which is a third very important way in which a community can be quantified. Right. So phylogenetic diversity typically, let me minimize a little bit more. Yeah. So a phylogenetic diversity is the diversity of all the lineages found in a community, right? So I think everyone here is a little familiar with what a phylogenetic tree looks like. It's the one that I've shown here in the right side, right? But it can also be shown in a circular representation. So it's like a dendrogram or a cladogram where the different species or any taxonomic level is connected to each other, and the distance between two species tells you how closely or not closely related they are, right? So here, if you look here, this is a phylogenetic tree of all the orders of birds. Now, the parrot community, the community A that I showed earlier, all parrots belong to a single order. And I think my highlighted rectangle has moved, right? So it's the Psittaciformis order, the second one from top. right? Whereas the community B, which had many different birds from different orders, would actually be found across this entire tree. We had owls, we had pigeons, we had parrots, and we had other shorebirds and waders, right? So they are found across this entire phylogenetic tree. So I can now compare those two communities in terms of what is the diversity of lineages which is found there. And the rationale here is that if all birds in a community come from a very small, a single or a subset of lineages, it means they're automatically performing fewer functions, right? Because functions evolve with time. So the farther apart two species are, the more distinct their beak sizes will be, right? So functional diversity and phylogenetic diversity are actually related to each other. The assumption being that closely related species also share functions. And we can see that on this tree also, right? So we have parrots are here, and for instance, we have galliforms here, right? So you have these birds which are very distinct from each other on the phylogenetic tree, and they are also functionally very distinct. The reason we use phylogenetic trees sometimes to measure diversity is because it is not always possible to quantify all traits or all functions in the wild of birds, right? I can measure beak sizes from museum specimens, I can measure body mass, but there are many different attributes of birds that are important functionally that I can't measure. So I use a phylogenetic tree as a surrogate for all the functions. Right. So now if I go back to that same distribution, so I've just shown here how phylogenetic diversity will change. And this is not the parrot example, but let's say we have, this is a small hypothetical phylogenetic tree shown here in black. You have two different clades here, 1 and 4. In clade 1, I have two hypothetical species, D and E. And in clade 4, I have A, B, and C. Each of these species is separated by a certain amount of branch length, right? And this branch length can be thought of as the number of mutations that are separating two species. So the farther apart two species are, the more mutations will accumulate between them over time, right? That is how speciation also happens very simplistically. More mutations accumulate and you keep on diverging. So if I have now from this simplified hypothetical phylogenetic tree, if I take two communities A and B, let's say I have a community A where I have A, B, C species. All three come from a single lineage, which is this lineage designated by 4. So the phylogenetic diversity of this particular lineage will be 1 plus 2 plus 5. Okay, it's the total number of mutations that have accumulated, or the total branch length that we call. Whereas a community B, which is composed of C, D, E, will be equal to 1, which is here, then 4, then this one, 5 plus 3. So all the branches or all the mutations that separate them. So phylogenetically, B is more diverse than A, right? And this is the same scenario that you will see if you compare A and B, where A is a community of parrots and B is a community of multiple orders, because B is more spread out along a phylogenetic tree. So automatically the sum of the branch lengths across the species of B is going to be much higher than all the parrots which are present in just one lineage, right? So you can have two communities where the taxonomic diversity or the species diversity is identical. They both have 20 species, but functionally they can be very different. You have a dispersion of 5 in beak sizes, but 15 for the second community. And phylogenetically, they are even more distinct, because when we consider functional diversity, we consider only 1, 2, or at the max 5 traits. But phylogenetically, all traits have contributed to them diverging. So they are more distinct phylogenetically, right? So this is the rationale behind the three kinds of diversity metrics and the three ways in which you can quantify or describe or compare two different communities. Now, how does all of this tie back into my community assembly? This is the important part, and this is very interesting, at least to me. I hope to you too, right? So, coming back to my original community assembly process, right? So, I said you can have this whole regional species pool, which are all the species found across an entire elevational gradient or a mountain system. Some species will be filtered out by the environment, the others will be filtered out because of high competition between them, and the resulting is what you observe, which is the species that are actually found at a particular elevation. Now, you can actually compare this local community to the regional species pool and then comment on the kind of process which has given rise to it or which has resulted in it. And this is actually something that we can see here. Instead of a regional species pool, you can think of a regional trait pool. We can go back to our beak size example. So if I have the beak size of all the birds found across all elevations, and I have the beak size of birds found at a particular elevation, I can say that some of these beaks are because they are environmentally favorable to that location, and some of them are going to be a little different from each other because if they are exactly similar, then they would outcompete one another, right? One would lead to the local extinction of the other one. So your local community can be viewed in terms of the species or in terms of the traits. uh, and same for phylogenetic, right? So you will have local community, which is evolutionarily a subset of the regional phylogenetic pool, because not all of them can be exactly similar, but they have to be a little bit similar to each other because they have to adapt to the same environment found. Now, how do you actually compare your local community to the regional pool? And this is where it all sort of ties together. So on the top, what I've shown here is a hypothetical phylogenetic tree, which is a tree of all the species found across all elevations, right? So this is your regional phylogenetic tree. It is also your regional trait pool because the different species have different traits. So I have 11 species here, which are found across 3 different clades or 3 different lineages, if we may. And each species has a different trait, which is given in 11 different colors. Okay, so we have 11 species, 11 traits, which are 11 colors, and 3 different clades or 3 different lineages. Now, if my local community, the one that I am actually observing and measuring, if it has traits which are all very close to each other, they have all come from one clade of the regional trait pool, then I can say that that community is dominated by the environment because the environment has strongly selected very similar traits that are all locally adapted. On the other hand, if you look at this community C, if my local community has 3 species which all come from different clades, which means they are very distinct from each other, I can say that environment has not played a stronger role, but competition has played a stronger role because these species are very distinct from each other. They will only be distinct if they are trying to minimize competition. So when environment selects for something, it selects for similar things because the environment is one, right? But when competition selects for something, it selects for distinct things because competition is trying to minimize resource exploitation from each other, right? And you can have a scenario in between where you have both of these processes acting, or you have something which is random. You can't really, it's neither very clustered on one clade, it's neither extremely dispersed, it's something in between, right? And this is how you can actually study communities in nature, by looking at either their traits or their phylogenetic relationships, because you can measure every single one of these things, right? You know exactly the kind of birds that are found in eastern Himalayas, you know exactly the kind of birds that are found at a given elevation in the eastern Himalayas. So you already have the regional trait pool, you already have the local trait pool. All you have to do is now compare the two. Where have they come from? Are the traits spread across the regional trait pool? Are they coming from a very small part of the regional trait pool? And that is exactly what we did in this particular study. I should have actually said that this work was a part of my it started during my PhD, but some parts of it we carried forward, and then I was able to do extend that work a little more, and this work actually came out very recently. So coming to the actual, so till now I was actually just talking about the larger field and how to study processes and quantify communities. But coming to the actual case study where we collect data. Uh, yeah. So what we did was we asked a very simple, seemingly simple question: what process dominates the community assembly of East Himalayan birds, right? And the process that we were interested in was environmental filtering versus interspecific competition. So this particular location is Eagle Nest Wildlife Sanctuary. It's in the West Kameng district of Arunachal Pradesh. It's one of the most species rich places on the planet. So here, if you see these two panels, these are the species richness density map. I think the one on top is the richness of endemic species or the range restricted species, and at the bottom is the overall species richness. So Arunachal is right here, this yellow orange part. So it's actually very high in species richness, but one of the highest in terms of the endemic species, which means the species which are found here are actually not found anywhere else in the world. So it's very important ecologically to study why this region is so high in diversity. And that's exactly the question we were interested in. So inside West Kameng district at here, there is a small wildlife sanctuary, just about 220 square kilometer, called the Eagle Nest Wildlife Sanctuary. It's in this white border. And across Eagle Nest, just sort of intersecting it, is this beautiful orange colored road. It's actually a motorable road, a dirt track, which crosses the sanctuary, but more importantly, it actually rises from just the southern border of the sanctuary, which is close to Assam, all the way to the highest point, the ridge line, which is close to 2,800 meters. So this road goes from around 500 meters to 2,800 meters, and it covers this distance in a matter of just 20 kilometers in a straight line, right? So it's a vast elevational gradient in a very small geographic space, which gives you an excellent gradient of environments to study how environment determines the composition of local communities. So we used this particular system, and this study, the field work for this study was done by Rohan Pandit, who is this man in the middle here. Rohan is an excellent bird watcher, but an even more excellent, even better person and an amazing photographer also. So a lot of bird photos that I've used here were clicked by Rohan. So Rohan did a remarkable job of collecting data on the local communities of birds. So what he did was he walked along this orange road that you see, up and down for almost 4 years, month after month, during summer and winter both. And he noted down the birds that are found at every 50 meters along this road, 50 meters elevationally. So he did this from 2011 to 2014 during the breeding season and also in the wintering season, from 200 meters to 2,800 meters at every 50-meter resolution, right? So we have a very good data set that tells us how our local communities changing with elevation along a very broad elevational gradient. He recorded almost 15,000 birds in this is just the breeding data. So 245 species in just between April and July. And I've not included here, but there was also a wintering data of close to 19,000 birds spotted in the winter. So it's a remarkable data set. And almost like a natural ecological laboratory for us to ask how environment determines the community assembly of birds in this region. So with this data, we were interested in asking how the community assembly processes change with elevation. We assembled functional and phylogenetic data for these birds. So we assembled these 8 traits from various sources previously published as well as measured from the museum by others, not by us. So we assembled data on wing length, tarsus length. Tarsus length is the length of the leg in the bird, which determines what kind of food it will acquire, but also the kind of habitat that it will forage in. Body mass, which is a proxy for how much resources a bird will use, but also for other life history traits. Beak size, that we have already talked about. So the first 4 are, of course, morphological traits. They determine in some way the kind of environment a bird will be able to live in, the kind of dispersal ability it will have, the kind of resources it will need. And the 5, 6, 7, 8 are sort of behavioral traits, which also tell you something about how different species will compete with each other in a given environment. So they are all different axes of functionality. Some of them are related to resource acquisition, some of them are related to resource competition, and others, right? So we gathered data on all birds, these 8 functional traits, and we also were able to obtain a phylogenetic tree for these birds. And which is why I love working on birds, because even though my primary interest is usually insects and moths, birds just have this wealth of information available for someone who's interested to work on them, right? If you can collect very good primary data, which is field data, in terms of everything else, there is a lot of information available to support your research. So this is a phylogenetic tree of 245 species that we were able to obtain from published literature, right? And what I've done here is just to show you as an example, I have actually colored the branch lengths by their body size. So red color means larger bodies and green is smaller bodies. So you can already see how species which are closely related are also similar in body sizes, right? So does not matter large or small. What matters is how colors are clustered across the phylogenetic tree, right? So it shows you how functional diversity and species diversity are usually very related to each other. Right. So what were we interested in looking at? Yeah. So here we actually used a existing uh, analytical framework, something known as the stress dominance hypothesis, which is very intuitive if you think about it. It says that any community of, does not matter only birds, any ecological community which is found in stressful conditions. In this particular case, we are talking about birds along an elevational gradient. So a stressful community is the birds found at higher elevations, because they are under sort of stressful environments. It's very cold, resources are limited, the winds are sometimes very fast, and they have to adapt to all kinds of rapidly changing wind and temperature conditions. So a community which will be in a stressful environment should actually be shaped more strongly by that stress. The environment will determine what kind of community can or cannot live there. Whereas a community which is found at lower elevations, that is benign or stable conditions, those are the communities which will actually have competition with each other, because there are so many, the resources are plentiful, the species have had the time to have larger population sizes, so resources become limited for them and they start competing with each other, they specialize more and more, right? So this is a nice framework which actually gave us something to something to test in our system, because we already have this stress gradient from low elevations to high elevations. We are studying the role of environment versus competition, so it's nice to have a hypothesis to actually test. So the hypothesis that we came up with was that communities found at the highest elevations will be dominated by environmental filtering, whereas communities which are found at the lowest elevations will be dominated by interspecific competition. So this is what we were interested in doing, right? So we collected, observed birds at different elevations for, and each of these community is now a local community For each local community, we measured traits and the phylogenetic diversity, which is the total branch lengths in that particular community. And we are now interested in seeing how the diversity, let's call it dispersion for now, right? Because higher dispersion is more diversity. How that dispersion is changing along the elevation. And we hypothesized, if you see in this bottom graph, we hypothesized that the divergence or the dispersion will actually decrease with elevation, because communities at the higher elevations are supposed to be more clustered or more similar to each other, the species, because they are selected by the environment, so they should have the same kind of traits, whereas at lower elevations they will be more different from each other because competition plays a very strong role. So competition separates them in terms of their traits or lineage diversity. So this is the pattern that we were interested in seeing. And what did we finally find? Yeah. So this was our hypothesis. Low elevation communities should be more dispersed than high elevation communities. And of course, I went a step further because we had summer as well as winter data. I extended this existing hypothesis, stress dominance, to seasonality. And I said that yes, okay, low elevation communities should be more dispersed than high elevation, but winter should be more clustered than summer. It's the same rationale that I am using. I am saying that winters are more stressful as compared to summers. So if I compare the communities across the seasons, I should see that winters have a more clustered because environment is stronger, whereas summer should be more dispersed because competition is stronger. So these are the two hypotheses that I am testing. Summer communities should be more dispersed than winter communities, and high elevations should be more clustered than low elevations. And what did I find? Lo and behold. So I actually found a very strong and remarkable pattern. So each dot here that you see is a local community, right? It's a community of birds, and each two consecutive dots are separated by 50 meters in elevation. And when I plot the functional diversity with elevation, I see that it steadily decreases, right? Which means, in terms of just this top panel, which is functional dispersion or functional diversity, I'm using them interchangeably for now. The communities found at lower elevations are more functionally distinct from each other, right? Which means competition is more important. Whereas at lower elevations, they are very similar to each other, the species, which means the environment has actually selected for very similar traits, right? And it helps to think of these in terms of a phylogenetic tree. It shows that the species within this particular community at the highest elevation have come from a single lineage or a very small subset of lineages, which means they all share very similar traits and very similar functions. Whereas that ones at the lowest elevations are functionally very dissimilar or over dispersed or diverged from each other, right? So this gives us a very nice result in terms of uh, how birds along such a vast elevational gradient are not just randomly found across that gradient. There is a very strong deterministic pattern in terms of which bird will be found where. And that pattern follows a very neat uh, analytical framework. It fits within this framework that stressful environments select for similar kinds of traits and similar kind of functions, right? So this was the pattern that we got across an elevational gradient. But like I said, I had also compared the patterns seasonally, and this is same thing that I found. When I look at summer versus winter in terms of functional or phylogenetic, summers are more dispersed. They have higher diversity, or they are more distinct from each other as compared to winters. This also matches our hypothesized expectation, right? So in terms of both the season and the elevation, you have this expectation and you have a result which matches that. Uh, I should probably skip this, but so what we asked was for each individual trait, what does the pattern look like? But I'll probably skip these two. Yeah, and I'll go here in the interest of time, right? So we asked a question: what process dominates the community assembly of birds in the eastern Himalayas? And we found that the answer is not very straightforward. There is no single process that dominates. The process which will describe or determine a composition, a community assembly, will change with elevation, it will change with season, and it will also change depending on the trait that you are looking at. We found that in some cases the beak sizes were more decreasing in nature than the body mass, right? That's the part that I skipped. But this basically tells you that community assembly itself is a dynamic process. Communities are not static objects, but there is a way in which you can make sense of those communities by looking at them along a continuous gradient of something. A pattern starts emerging when you compare different communities along a gradient. It also tells you that biodiversity is not just species richness, it's not just species diversity. You get different pictures and different stories coming up when you quantify biodiversity using different metrics such as traits or functional diversity and lineage diversity or phylogenetic diversity. What we are doing now actually is we are repeating this sampling, and I'm very excited about this. So this is a work that we'll hopefully start very soon. So we are going back to the exact same place and we are repeating our sampling strategy again after 10 years. It's actually a little more than 10 years now. We'll again go to the same road. We'll again quantify birds found at every 50-meter elevation, and we'll try to compare the pattern, what it looks like now. Not just along the elevation gradient, but for the same community across 10 years, right? And this will tell us something very remarkable about these communities because over time, at the same place, if the role of environment has increased, it makes those communities very vulnerable to changing environment, right? Any community where environment dominates the assembly is very vulnerable to environmental changes, and which is why along tropical montane systems in general, your highest elevations are the most vulnerable to fluctuating environment because the species there are highly adapted to those specialized habitats, and any fluctuations can actually have cascading effects across trophic levels, right? So yeah, so community assembly processes tell us not just about who is present, but also about the vulnerability of that system to future perturbations. So hopefully in the next iterations of NPTEL, I will be able to share some of these things coming up. How have the communities changed over time? But what I want to emphasize for now that this is not just about birds on a mountain, right? It tells you so much more about a system and how it is vulnerable to all kinds of external changes. Because what I've used here is actually just it's a model system along a stress gradient, right? It's an elevational gradient, but you can think of any stress gradient. You can think of a pollution gradient along a shoreline, right? You can have a pH gradient along a lake. You can have a disturbance gradient from forest to urbanized. Any gradient where you have stressful conditions on one end and benign conditions on the other, you should see this pattern, right? And if you see more and more the stress increasing more and more across that gradient, you will see the role of environment increasing more and more. So it's a very nice analytical framework to test a lot of things about communities and nature. Uh, yeah, and finally, I want to thank all the people behind the scenes. My PhD advisor was Ramana, who is also part of the collaboration going forward in terms of repeating the transits. But then, of course, all the wonderful people in the field without whom this work would not have been possible, especially Raju and Bayum and Vicky. Uh, yeah, and I think I finished just in time for the questions, Amrita.
Yeah, yeah,
Thank
yeah.
you everyone for listening patiently.
Thank you, Mansi. That was wonderful talk. So I'll paste the questions.
Yeah.
Sakshi. So Sakshi has asked. So I think this is watching a previous video, but she's attending the video live now. So, anyways, I can read out the question for you. So, what does over dispersion mean here? Or what does clustering mean in this context? And you have mentioned that the strength of the environment filtering is decreasing with elevation, but shouldn't it be the opposite with SDH? That's what she said. So she's confused with the
Okay.
terms. Yeah,
Yeah.
dispersion.
Okay. Right. So what does dispersion mean here? I'm sorry if I was not clear. So dispersion means what is the variation in something, right? So the more distinct your beak sizes are, for example, the more dispersion there is, the more variance there is, right? So when environment selects for something, it selects for similar things. It because the environment is selecting for a particular kind of trait, and which is why your dispersion decreases when environment selects for something. But competition minimizes dispersion, maximizes dispersion, because competition selects for distinct objects, so it increases variation and increases dispersion. So which is why along an elevational gradient your dispersion goes down, because environmental strength increases, so variation decreases. I hope that answers both of them.
Yes. Uh,
It does.
yeah.
I have a follow-up.
Yeah.
So in the slide that you showed today, the two negative slopes for functional diversity and phylogenetic diversity. So both had negative slopes and both had uh, um, clustering towards higher elevation, right?
Yes, yes. I think you're talking about this one.
Yes, yeah,
Yeah,
exactly this one.
yeah, right,
So can you just explain like how is SDH as a hypothesis being proven by these two graphs?
right, right. So stress dominance hypothesis says that in stressful conditions, yeah, I was not very clear about this. In stressful conditions, the environment is going to select for very specific kinds of traits which are very well adapted to that environment, right? Which means there are going to be certain species all very similar to each other which will dominate that community, hence stress dominance, right? So in stressful conditions you will have fewer dominating species which are going to be very similar to each other. Because they are similar to each other, it is going to be under dispersed, which means low variation, which is why your dispersion is decreasing with elevation. High dispersion means more dissimilarity,
Right.
means the species are more different from each other.
Okay.
Does that help?
And does it also have something to do with the fact that the phylogenetic diversity slope is greater than the functional diversity value slope?
Uh, yeah, so this is some of the things that I want to deeply look into, because when you quantify functional diversity, I used
Mm.
8 traits, right? But I'm sure those birds have so many more behavioral attributes that I was not able to quantify, but which the phylogenetic diversity captures, right? Because it's the sum total of everything, right? It's the difference in their DNA, which means it encapsulates all the things that I have not measured also. So, which is why you see a stronger response when you quantify phylogenetic diversity. And functionally, it is actually just a subset of this because,
Right. Okay.
right? Yeah. Does that make sense?
Yeah, it makes sense now. Thank you.
Yeah. Thanks, Mansi. So there is another question in the live chat. Since Eagle Nest is so geographically compressed, only 20 kilometer, do you think mass effects from lower elevations are blurring the SDH signal and inflating in diversity at the top? This is from Mania. Yeah.
Uh, that's an interesting question. see two things here. One, yes, it is geographically compact, but it is environmentally extremely diverged, right? So yes, it is only 20 kilometers. So for a very mobile animal like a bird, which can easily just fly from one community to the next, even in a community like birds, which is highly mobile, we see this strong pattern, right? We see a community which is at which is at 200 meters versus 250 meters, different from each other. See the trend line, right? Communities are rapidly changing, which means birds are not really flying around. Uh, is mass effect from lower elevation blurring SDH signal? I see a very strong SDH signal, right? So mass effect is not blurring it because I see it. I can see this trend line with a strong negative significant slope. You will not, of course, see every single, you will not see a slope of -1 with the communities neatly along the fitted line. Of course, there will be some noise in it, but the mass effect, though I have not explicitly tested for it, if I have to speculate, I would say that it is not acting up here because the communities are really changing very rapidly. There are no birds which are found across the entire elevation. The birds which are found at lower are not even found at, you know, low mid versus mid. Forget about high. So the communities are changing very rapidly for birds. And to give you context, I study moths also in the same region. So there is a particular group of moths called hawk moths, which I had studied extensively. And for when it comes to hawk moths along the exact same transect elevational gradient, there are at least 25 to 30 species which are found across the entire elevational gradient. So a species which is found at the top is also found at the bottom. It's almost 70% of the communities are similar, but that's not the case for birds. Birds are extremely specialized in this region, very locally adapted. Yeah.
Yeah, and Shruti have asked about Pagel's lambda. Could you please explain the Pagel's lambda in your experiment and the significance?
Right. Okay. Fine. So there was a particular phylogenetic tree that I had showed, and I thought Pagel's lambda was actually cropped, but oh, it must be in one of the previous slides. Okay, fine. Yeah. So it's actually a metric that you use to quantify the degree to which a particular trait is distributed along a phylogenetic tree. So if I can use the example of this particular graph that you see here, which is a phylogenetic tree of 245 bird species, and I have colored the branch lengths by using the body size in this case. So I can see when there is a pattern where colors are clustered across lineages, it means the Pagel's lambda is high, right? If I see a pattern where there would be one red here, then one red here, one here, it'll be like sort of, randomly distributed across this tree. That would mean low Pagel's lambda or low tendency of that trait to be conserved within a lineage. That's a little rare in nature because closely related species do tend to be similar, but you can have scenarios in which traits just sort of independently evolve across lineages, which is what happened with flight. For instance, flight evolved independently in the mammalian lineage for bats. It had nothing to do with the existence of flight in insects or birds. It was, so these, if I look at the evolution of flight across insects and mammals and birds, that would be a low Pagel's lambda. That trait has nothing to do with the phylogenetic proximity of these three lineages, right? But when I'm looking at just birds and body size, I can see that closely related species tend to have similar traits, which is how something can be quantified using Pagel's lambda.
Yeah. So we have one more question. Did roadside bias potentially influence the trait data, even though the team used the sound data to detect the birds deeper in the forest? That's
Right.
Mm.
Yeah. So the traits were actually not collected by us. It was published literature. So there are a lot of databases available for traits, which are measured from museum specimens or sometimes in the field from other parts of the world. But mostly in Arunachal, people have done the work. So I doubt that the roadside bias would come in the picture.
And what would be the reason behind such difference between the bird communities which are bigger in size, thus more mobile versus the community of moss, which would require more energy and adaptations perhaps to move across such elevation? Yet we notice that the smaller animal is more adapted to live across a broader elevation spectrum.
Yeah. Yeah, that's a very interesting question, and I hope to figure out the answer soon and tell you. But for now, I can tell you something that the moths that I studied were just one family of moths called hawk moths, which are highly generalized group when it comes to moths. I'm my bet would be that the pattern will change as soon as I consider all moths, right? So I'm considering all birds, but I'm not really considering all moths. And the moths that I did look at are very aerodynamic. They are spread across the entire globe, not just in this region. So it's not really the best comparison to make. And the only point that I should probably making is how patterns change from group to group. Yeah, taxa to taxa.
Yeah, thank you so much, Maxi. I think today we can wind up the session. So if students, any of you have any doubts, please write to me or you can post a question in the discussion forum. So one last question from Debelina. Do you have any bird communities which live across these elevation?
Yeah, I think you mean species which live across these elevations. So there are a few warblers which are found across almost 70% of this gradient, but we really don't have a single species which is found at 2800 meters also and 200 meters also. The birds are really range restricted, and which is perhaps why this region is so high in endemic diversity. These birds are really specialized. We have the Bougainvillea cicla, which is found in very small patch.
Yeah. So, students, like you should search in Research Scholar, I think ResearchGate or Google Scholar. So all of Mansi's research works are there. Yeah. And if you have any doubts, please post in the discussion forum. I can get a response from Mansi and post back. Yeah. Thank you so much, Mansi, for the wonderful
Thank
talk.
you.
And thank
Yeah.
you everyone for joining in. Yeah. Thank
Yeah,
you.
thank you.
Thanks,
Thank you.
everyone.
Thank you.
Yeah. Thank you.
Just one comment I would wish to make.
Mhm.
Dr. Mansi, this is a
Yeah.
beautiful talk. The
Thank you.
language mastery, what you have, amazing. And
Thank you.
you used about 2 to 3 dozen very most appropriate words, but very rarely used words. You placed it
Oh, thank
so
you so much.
beautifully, so naturally. It's a beautiful talk. Very nice.
Thank you. I'm glad.
Yeah,
Thank you so
and
much.
that shows that how much you have just got into the whole thing, like in your research, your
I'm pleased
activity,
that I
and
was
this
able
thing.
to convey my understanding
It
properly.
was, see, so most of the many, at least, see, for me, I am beginning with the many of the concepts new, at least about 20 to 30 definitions, what you mentioned. You
Yeah.
place them with such clarity. Amazing. Thank you very much.
I'm glad.
Yeah.
Thank you so much.
Yes, thank you.
Thank you. Yeah.
Thank you, Mansi.
Thank you, Amrita.
Yeah,
Thank you. Bye. Bye, everyone.
Yeah, bye. So we will see our next weekly live session with Dr. Umesh Srinivasan and Dr. Robin. So this happens 27th Friday, 5 to 6 p.m. Yeah, thank you, everyone.